Welcome to the Global Harmonization Initiative webinar series. I'm Dr. Diana Bogieva, President of GHI, and I'm thrilled to have you here. Our exciting webinar today explores the realm of potential toxic compounds in food. This session will address two critical topics. Specifically, we will explore oxidation products in plant oils and the presence of inorganic arsenic in rice. Firstly, we'll discuss the potential oxidation products in plant oil, and after that, we'll talk about inorganic arsenic in rice, or vice versa. Get ready for an informative and engaging session that will broaden your knowledge and perspective on potential toxic compounds in food. But before we start, I would like to introduce two announcements. The GHI World Congress scheduled from June 25th to June 27, 2025 at Hilton Rotterdam in the Netherlands will soon begin accepting abstracts and registration on its website, which is set to launch in the first week of May. We are eagerly anticipating the exploration of the Congress team connecting food security, safety, health, and sustainability challenges ahead. Keep an eye out for the Congress website, which will be live and accessible very soon. Stay tuned for more details. Additionally, regarding our webinar series, including today's session, I'm reminding you that all webinars will be showcased in the GHI YouTube page. To stay updated, ensure you subscribe to our channels. You'll receive notification whenever a new webinar is uploaded, guaranteeing you don't miss valuable insights and discussions. Back to our webinar. We have our special guest, Professor Dr. Jörg Feldmann from University of Graz, Austria, and Dr. Associate Professor Michael Markovic from Graz University of Technology, Austria. He is also a co-chair of the GHI Chemical Food Safety Working Group and a chair of the GHI Supervisory Council. Before we continue, I would like to say a big thanks to ICC providing us the system for these valuable webinars, and also big thanks to our GHI Communication Director, Nicholas Stanley, and our GHI board member, Dr. Gerhard Schlenning, for their big help and contribution to these webinars. Let's start with our first guest speaker, Professor Dr. Jörg Feldmann. He is an environmental analytical chemist who did his master's in geochemistry in South Africa and a PhD in environmental analytical chemistry in Germany. He also spent 2 years at the University of British Columbia in Canada as Alexander van Humboldt Fellow, before spending 23 years at the University of Aberdeen in Scotland, UK, and then moving to Austria in 2020. He has given around 200 invited presentations and published more than 300 peer-reviewed papers and received numerous prizes and medals. More info could be found at the GHI website. We are very excited to have you with us, Professor Feldmann. Over to you.
Okay, thank you very much, Diana, for a very nice introduction. Okay, so yeah, I would like to talk a little bit about the arsenic in rice and into the legislation, and what comes next, really, because I think the story has not an end, as usual. So in the next 30 minutes, I would like to give you just some introduction into arsenic a little bit, into other things, and then basically state where we are right now and what we will probably do as a community in the future. So just to my background, as you have heard, I'm interested in I'm an environmental analytical chemist interested in toxic elements, And you can see in the periodic table, there's a lot of them. Usually we work on mercury and arsenic, selenium, cadmium, and recently quite a lot on fluorine in form of PFAS. But this is not what I would like to talk about here today. Today we focus just on arsenic. And when we look at arsenic, one word always plays a role, and that's speciation. When you look it up, what means speciation? I thought I define this before we start, because people understand different things about it. So speciation, when you look it up in Wikipedia, then it basically in biology, it's an evolutionary process by which population evolved to become distinct biological species. And the most cited scientist in that is Charles Darwin. That's not what I meant, and not what I mean by speciation. We in chemistry define this speciation analysis as the analytical process to determine the molecular form of an element, so chemical species. So basically, when we talk about arsenic, we are not talking about just arsenic, but what kind of molecule we are having here. Why do we do this? In general, chemical species of an element have different toxicities, solubilities, bioavailabilities, and reactivities. And understanding speciation of an element means we understand its environmental and biological processes and also its toxicity. And this is important in terms of food safety, for example. So after this short introduction into arsenic in food, I will come on directly or indirectly to inorganic arsenic in rice, a question about speciation analysis, and then what is next? There are some new arsenic compounds which we should tackle worldwide to make food more safe. So arsenic problem, we have a worldwide problem. Everybody probably knows about this. Arsenic is toxic. You can kill somebody instantaneously with it. But arsenic in low concentration can cause cancer as a class 1 carcinogen, as we know, and it's very much regulated in water, in drinking water for a long time. And this is here on the graph. These are the cancer slopes from the US EPA. So different types of cancer correlate very well with the inorganic arsenic ingestion, milligram per day, for example. And then you can calculate the excess lifetime risk to contract cancer. These are the cancer slopes, as we know. And then on that basis, the WHO, the US, EU sets the levels to 10 ppb or 0.01 milligram per liter arsenic in drinking water. When we started the work in 2002, there was nothing about food regulation. So your food could contain as much arsenic as you want, and it was still fine on a market. And then discussion started actually so in 2005. So no regulation in 2010, for example. So what we have done over the years, over the last 20 years, we looked at the whole process, and we're interested in rice production. So we look what is going on in the soil, what is going on inside the plants, the different varieties we looked at, how is it translocated, the toxic compounds into the grains, and what do we do with the rice? So in terms of cooking and things. So the whole bunch. So from farm to fork, that's what we are interested in. And with that, that was a time when I was in Aberdeen, and I worked a lot with Andy Mihak here, who is now in Belfast, and we have done a lot of work together here. And then here one snapshot, basically looking at geographical difference in total arsenic in white rice grain, It's a global distribution. You see here from Egypt up to US. You see there's a huge differences here, and here USA gets a gold medal for the average highest concentration of arsenic in rice, and that was in 2008, 2009. Approximately. So what does it mean? Well, at that time, there was only a Chinese import regulation for inorganic arsenics there, and that was 0.15 milligram per kilogram, and that's inorganic arsenic. So here you can see that a lot of these rice samples on the world market are above this limit, and they need to do speciation analysis and then maybe cannot be exported to China. So that is basically a quality control here. What is a problem with arsenic in rice and in also other food is, in contrast to water, why do we have no regulation in food? The reason was basically arsenic occurs in different molecular forms, and we know only that inorganic arsenic, this is basically arsenate, so the pentavalent arsenic, and arsenite, the trivalent arsenic, considered as a class 1 carcinogen together, and in drinking water we have only these compounds really there. Therefore you can do total arsenic analysis. But in rice we have also these two fellows here, so the dimethyl arsenic acid and the little traces of monomethyl arsenic, and they are considered, well, as co-carcinogen, but in general as non-toxic. So if legislation goes ahead with a discussion 10, 15 years ago, then it should focus on the class 1 carcinogen inorganic arsenic. The trouble with arsenic is that it occurs in a lot of different compounds, and inorganic arsenic can occur in these two different forms, but in legislation is considered only both of them, then they can easily be transferred analytically, so that arsenic 3 can be oxidized to arsenic 5 by purging, expose them to hydrogen peroxide. And when you do, you can extract them by acid quite nicely, and these acids do not destroy the other species, so no transfer of these species to those compounds take place. And so the method of choice in the EU, and when it was discussed first of all at the WHO and the US FAO, is basically the recommended method here, coupling an HPLC or an anion exchange chromatography with an arsenic specific detector, and this is an ICP-MS. And when you do that and you have an extract of, well, nitric acid with hydrogen peroxide, you get a chromatogram like this. So you have a lot of organic compounds, but you only detect the arsenic compounds in here. So you have your dimethyl, your monomethyl, and then the inorganic arsenic as arsenate here. You see a chromatogram of a certified reference material down here. The detection limits are very nice. And this is very specific, and so that you can actually then quantify the inorganic arsenic quite well. So do we actually need to quantify it, or is the ratio of these species in the rice always the same? And when you look at this, these are from also an old paper, basically looking at rice varieties in Bangladesh, China, India, Italy, and US. And you can see here that the total grain arsenic versus the inorganic arsenic shows different slopes. Here that means we have different ratio of inorganic arsenic to total arsenic, depending where the rice is coming from. We have regional differences, for example, seeing here also as DMA. And what it means is basically total arsenic does not reflect toxicity in the rice. Hence, inorganic arsenic needs to be determined. Sad, but it is. So and that is a very complex method. HPLC or ion chromatography coupled to ICP-MS. Only a handful labs in the world had this at that time. available, which is not good when you want to control food safety. So, but having, if you do that now, and want to do a risk assessment, so we basically took the daily consumption of rice per country. For example, here you see Bangladeshis have 450 grams per day, whereas here in in Europe we have maybe 20 to 50 grams. And if you multiply this by the inorganic arsenic content of the local rice, then you come to the daily intake of rice. And when we apply the US cancer slope, which I showed you before from the water, because we know that the arsenic you eat with the rice is total bioavailable, then you can actually calculate an excess cancer risk per 10,000, and you can see that there are quite high cancer risk. Usually acceptable is 1, and therefore you can see by just eating rice what the average person does, it would exceed the acceptable excess cancer risk by eating just rice. And we thought this is basically a good case, so that people are like here in Europe, the EFSA it's a European Food Safety Authority are looking into this and maybe discussing the maximum level of arsenic in food. But they basically say, well, this is not clear, there's not enough data there, and nobody can do the analysis. Basically, they blamed the analytical chemist for not doing, coming up with maximum level. And that was the same response was on a WHO and the US FAO meeting. And therefore, it was a need from the analytical community to show that we are able to analyze this. So I spoke then to the European JRC group. This is basically the lab. They make standard reference materials to organize the proficiency test. And we provided the rice, and they processed them, and then they send it out to 100 labs worldwide. The rice, there was identification of maybe the expert groups, so we knew what we would expect. And 30 labs came back with also speciated data, and these are the data here, as you can see here. They are quite nicely distributed, and from that trial, which was then published in 2011, I would say the summary is the introduction of the maximum level of inorganic arsenic in rice should not be postponed due to analytical concerns. Basically, the analytical community has done the job, and then that was accepted then later on at WHO and FAO level, and then it was implemented into European law from the European Commission with these numbers here. So we have not one maximum number for inorganic arsenic in rice. We have a few of them, and one of the lowest is concerned about toddlers or infants and young children, because they have a more food intake per kilogram body weight than adults have. That's why a lower number is here. And you see also there are difference for milled rice and husk rice, because a lot of arsenic is in the husk or in the bran around the rice, and therefore there are higher levels. So, and a group here in Belfast, they checked basically, Andy Meag's group, they checked basically if the regulation was respected in 2016. So they collected baby rice, rice cereals, and rice crackers, and you see before introduction and after the introduction, there's no change. Basically, a lot of these rice samples are basically violating the new maximum level of inorganic arsenic in rice. and that is not good. What is the reason for this? There's enough monitoring is taking place because the food labs around the corner do not have the sophisticated HPLC ICP-MS methodology. If you can't control it, the companies basically are still exporting the rice into Europe or producing it in Europe. So what do we need is basically we have a problem, especially the food, baby food companies are looking for really for low arsenic rice, and they're lacking to monitor this. So we thought we have to simplify the sophisticated method so that we get some kind of a quick test, like a traffic light system, like what we all know from the COVID, the quick test we perform. And we wanted to also develop a test, and there's a lot of chemistry behind it, which is more than 100 or 150 years old. It is based on the Goodside method. Arsenic species, when it is inorganic, you can actually volatilize this to arsine by treating this with sodium borohydride in acid. And if you have organic arsenic, like arsenobeta or sugars or DMA, this is not generating volatile compounds if you have the right conditions. And MMA is often is absent in the rice or in seafood, so that can be neglected. So you can do what we call a hydride generation, and then just have a gas liquid separator, and the gases go up, and then you can use atomic absorption, atomic fluorescence, or ICP-MS as this. But we wanted to do it even much easier. We wanted to have a field kit, and basically where we can measure this really in a field with the rice farmers. Basically you scoop 5 grams rice, mortar it a little bit, boil it in nitric acid, and then pop in a sodium borohydride tablet, and then gases are generated. And here in the top filter, the gases are basically have a reaction with the filter is impregnated with dibromide, and when arsine is generated, that forms a colorful complex, and that's what you can see here on the filter paper. If you have more arsenic in there, then the color is more intense, and that's what we measured on 30 odd rice samples, and compared this with what we measure with the gold standard with HPLC ICP-MS, and you can see the slope is roughly 1, and it's a good and the error bars are quite small. So this is almost is better than we thought, and it's can be used as a traffic light system. And all the analysis costs not 100 or more dollars per sample, but maybe only 3 or 5. So it's much cheaper, and you can do it in a field without electricity. You can, and within 1 hour. And here we tested this. If they would violate the 200 ppb of inorganic arsenic in white rice, with the error bar attached to it, and you see from the 30 odd rice samples, 4 were above or around that level, as you can see here, and they were detected with HPLC ICP-MS as well as with the field method. Therefore, we have no false positive or negative to record. So a very trustworthy field kit, which can be used also in developing economies. So a very affordable method. And we try to use this in a country where no other facilities is really available. In Malawi, is one of my PhD students, Angston Langeni, was from Malawi, and we said, well, let's go down there and test out the system by going up and down the country at the Malawi lake here, going from rice farm to a next, and then do the test on site, and then take the samples back home and then measure it in our lab. And you see it's a very good correlation. What we did as well is then basically empowering people according to the SDGs, basically empowering people and basically training rice farmers or leader of rice farmer communities to do this kind of tests then on site, and they can then decide on to sell their rice products with very low inorganic arsenic, for example, to the European community. Did this survey, he had to stop at one point because he didn't have a nitric acid enough, and he couldn't get that. And we said, okay, we should find another worldwide a standardized acid what you can buy everywhere. And we tried a few things out, and it turns out Coca-Cola actually works quite well. We just recently published this, the detection of inorganic arsenic in rice using the field with cola extraction. I have to say Coca-Cola Classic only works. Zero doesn't work, it foams too much. But you can see here we had also another set of samples, and we had two false positive were measured in the baby rice samples, which is much lower than what we tested these things before. This is here from the Austrian market. So this field kit works quite well. And you can see here also the levels from these recent rice samples from the market shows that the level in the EU goes down, or it looks like that it goes down. But still, there is a need for really low level of inorganic arsenic, and we will see this new legislation later on. So there's a real need for low inorganic arsenic. And then I spoke to baby food companies, and they wanted that. And you can see here, Brazil would be a very good country, one of the biggest rice producers worldwide. You can see here in Brazil, they have only total threshold level of total arsenic for 300 ppb here. And you can see a lot of them samples are above that. But what we noticed is in Mato Grosso, that's very inland, the rice is very low. And the reason for this is upland rice. It doesn't grow in paddy fields, so underwater. It grows on aerobic soils. And that is why it is so low. And however, you might have then some problems there with aflatoxin, but the same as when you grow basically a wheat or barley. But inorganic arsenic is very low, and we can discuss this maybe later on why that is. We wanted to test now 5-6 years after the introduction of the law what happens on the market, and we did a rice survey in the Austrian market with the different maximum concentration levels of inorganic arsenic for baby rice, brown rice, rice crackers, and white rice. As you can see here, 50 samples, and a lot of samples need to be tested for speciation because the total amounts are very high. And just here, focusing on the baby rice, and what occurs to us is, first of all, almost all rice samples are not violating really the new laws, which is a huge development in the last 6 years. But what we noticed is that the law or the regulation is only about the inorganic arsenics. This is low, but in some baby rice samples, the DMA is very, very high. And this is somehow worrying. Although it's not regulated, so they do their bit, but it's a bit worrying. And why that is, just in a moment. And this is what I would like to tackle now, is basically what is coming next. Is basically, well, if you have DMA in there, that could be not in the form of dimethyl arsenic acid, but it can actually exchange the sulfur. Instead of an oxygen, you have here a sulfur. We call it thio-DMA, or sometimes it's called DMMTA, to dimethyl monothioarsenic. And this has been unnoticed for a long time, for the last 10-15 years, because we were all extracting the rice with acids. But if you use an enzymatic extraction, you can actually find these compounds. So what is the problem with this is basically that compound has been tested to be highly cytotoxic, and it occurs everywhere worldwide in the rice. So if you have high DMA, you have also high amount of this compound. So this needs to be looked at, and this is down to the work of Britta Planer-Friedrichs group and co-workers in Germany. They have done a few papers on this. The big problem is, for example, also rice cakes. So if you have DMA and you do a puffing of rice at a higher temperature, you generate these kind of compounds, and the cytotoxicity actually increase very highly. The problem now in baby food, I come to this, is now very, very low, and it's often full with DMA, and maybe especially when you have a temperature treatment there, you generate these kind of compounds. Rice crackers are full of it. Babies are often eating these rice crackers, but it's not declared as rice crackers. And this is a big loophole in the law. So we have to do some citizen science really there to get a hold on this kind of thing. The other, what is next, is basically the arsenic speciation is much more complex, and I talked about just here. When we go from rice to seafood, then we have a lot of arsenic in the form of arsenobetaine, which is absolutely non-toxic. But then we have in seaweeds a lot of arsenic sugars and arsenolipids. Most of these blue ones here labeled haven't been tested for arsenic toxicity, but here in the group of arsenolipid, and that could be 50 or more different types, 2 have been tested in the last maybe 5 to 10 years, the arsenohydrocarbon and the arseno fatty acids. And this is a little bit worrying, because what they found is basically in cell cultures. This is mainly Tanja Schwertler group in Potsdam, looked at bioavailability. That's highly bioavailable, these compounds. They go into the cell, they react with reduce the mitochondrial membrane, they disrupt neural networks, and they can enhance the permeability of the in vitro blood-brain barrier. So it's in the C. elegans, we looked at in vivo toxicity, and it shows that. So these are highly toxic compounds, and they are not regulated yet. So previously, we always thought if arsenic is in the form of organoarsenicals, they are not toxic. So they are like Dr. Jekyll. However, it turns out to be that they are also like Mr. Hyde. So why is that so worrying? There's not much done for humans, so some kind of feeding study, but we thought we're looking at mammals who eat a lot of seafood, and these are the whales. The whales here you can see on the pictures, they were stranded in Scotland, and we looked at them, and they are all age groups, and we wanted to find out how arsenic actually accumulates. And you can see, as we know, this is liver, kidney, muscle. It doesn't happen. But here in the brain, which is protected with the blood-brain barrier, the arsenic is actually increasing, especially especially in the developing brain. So this is a problem, and we we thought, well, arsenic is the brain is usually protected from the carcinogenic arsenic, but if we have lipophilic arsenic, it might penetrate, and we identified them by very complex analytical methods, so HPLC with ICP-MS and parallel with electrospray-MS. So we could identify these kind of arsenolipids in the brain of the whales, and we can see that they are basically penetrating through the brain. And this really asks the question: should we not do something here? What do we learn from this? If it accumulates in a whale brain, does it also accumulate in human brains? Well, we don't know, but there is a danger there that we maybe expose kids to arsenolipids when they eat seafood. What we know, This is a group here from my colleagues here in Graz. They looked at these arsenolipids and they identified them when women eat a lot of seafood, that it actually occurs in breast milk. So I think this is a battleground for the next research and try to get that legislated in the next maybe 10 years. And last but not least, European Commission updated really the regulation last year. And you can see we have lower levels now for polished rice, it lowered it, and we have new levels here for infant formulas that goes down to 10 parts per billion. Very, very low. Baby food, 20 parts per billion. Where can we find new rice which can actually not violate these laws? This is a big problem right now, but hands up for the EU to actually improve the quality of food in here. So take-home message from today's talk is that I hope I could show you arsenic toxicities dependent on speciation a little bit. Well, I didn't show this, but I mentioned it. As there quite often we have very complex analytical procedure for determining inorganic arsenic, but there are also some quick tests that are available and also in low economies that can be used. We have the last 10 years a first regulation of maximum level of arsenic in food, and especially inorganic arsenic, speciated arsenic in rice, and the rice content, the inorganic amount in inorganic arsenic amount in rice has been lowered. We see this in the market. We see, however, that also some baby food have, however, high DMA in there, so which is not regulated. So it's according to law, it's all fine. Then we have a few questions here for the EU regulation. Extreme values, low level values for baby food. Well, which rice is available? Do they use upland rice or not? And what happens basically to the rice which are higher? So I think it's absolutely necessary to monitor the inorganic arsenic in rice outside of the EU. If all the high arsenic rice is basically dumped outside of the EU on their market, which is not quite fair, as I would say. And on the new arsenic species, the thiodMA should be looked at. It's highly toxic. The rice cracker needs to be checked because they are fed often to toddlers, and the arsenolipids should be looked at. With that, and I would like to thank the people who worked on the arsenic in the last years, on some collaborators here, and I thank the whole team, the people who give money for this, and I like to thank you for your attention. Thank you very much.
Thank you very much, Jörg. This was a really, really insightful presentation. You mentioned actually arsenic toxicities depending on speciation. So which species of arsenic is more toxic?
Well, this is basically, if you look at food, then it's the inorganic species. Quite often there are some LD50s for arsenite. This is lower than arsenate, but when you eat it, it transfers to each other. That's why the legislators basically said we don't distinguish between them. We say inorganic arsenic. Even if you have thioarsenate in there, this is also would transfer to inorganic. So this is a class 1 carcinogen, and that is a focus of the legislation. But as I said in the last minutes of the talk, there are two compounds which are also highly toxic, which haven't been looked at very much by the environmental monitoring, food monitoring community, and this is thiodMA, and also which is also a metabolite when we eat seafood, for example, and arsenolipids, which is abundant not in rice but it's in seafood, and these are also not very well tested methods, only in cell cultures, but it seems to be that they are highly toxic. What we know only is the arsenobetaine, the major compound in seafood, arsenic compound in seafood, this is clearly not toxic at all. That is what we know. and the other ones have a question mark.
Yeah. So how much arsenic in seafood and rice should we consume during our lifetime to avoid the risk of developing cancer?
Well,
That's another question that
there's another
people have.
question. Yeah, no, thank you for the question. No, I think if you eat it in moderate terms, then it's fine. And there are some, as I said, if you live in the EU, any rice on the market seems to be that they are below the levels. Usually, I would say basmati rice from California is good. For example, from Italy is good. Usually, these kind of, and also from India, there are also low levels. How much can you eat not contracting? This is only a risk assessment. You can't say that you can get cancer. It's only a risk assessment that when you eat, I think 100 grams per day of rice, then you have a chance, I think 10 in 10,000 that you can get cancer from it. So it's relatively low. The biggest problem is if you live in a country or in an area where you have a lot of arsenic in the drinking water. This is much more of a problem. And then don't
Because
use
another
that
question
water.
for the drinking water is good that you mention it.
Yeah, okay, go ahead.
No, you continue with this. Just explain about the arsenic in drinking water, because this is another question that they have. People are most likely exposed to inorganic arsenic through drinking water, similar to eating rice. Or what country has the most issue with this? This is what the question of we have.
Well, yeah, yeah. If you are getting only your drinking water and that's high in arsenic, for example, in countries like Bangladesh, where the local maximum limit is not 10 parts per billion, it's 50 parts per billion, is already high. And there are a lot of communities, they drink water in Bangladesh, which is from shallow groundwater sources, which has maybe 100 and 200 parts per billion. And then the chances are much higher. So I've been a few times, a lot of times to Bangladesh, and I saw also patients on this. If you live in the area and you have access to arsenic, a low arsenic water, then drink that. But don't use also that water for cooking your rice, because then you get your arsenic in. There's also, if you are worried and you eat a lot of rice, then what you can do in general, you can actually soak it overnight and then decant the water. A lot of arsenic can be removed through that. It's not so efficient. As another efficient way is basically cook the rice with excess water, not basically boiling it down until no water is there, but excess water, and then you decant them. A lot of arsenic is then lost there. But of course, this is also a problem. Then you need more energy for this. This is not good. So there are different ways of doing this. But Bangladesh has a big problem. They eat a lot of calories, especially for the poorer people. They get a lot of calories through eating rice and nothing else. It's not very nutritious. Milled rice, so white rice. Then, and if you eat a lot of this, then and then you add extra arsenic from the water, then of course every food source increases the risk. One word to the white rice, to brown rice. We all should eat brown rice. People saying this, yeah. So, but you get the extra dose of arsenic in. So
brown rice or white
in brown rice, in brown rice, because it's in the bran and
in the bran
outside. And so bran is also something people are eating sometimes, and it's used, for example, in Africa to give children who are malnutrition, because there are a lot of nutrients in there. But we have to be aware of that contains also arsenic, so in a higher dose. If you deal with communities who have no other means of nutrients, then I think brown rice should be eaten, and despite the arsenic. However, if nutrients coming from other sources, then I would say get rid of the bran and eat the white rice for calorie
Yeah.
source.
You mentioned the methods of cooking. This other question that we have, what the consumers can do. You mentioned soaking the rice, but they said also, does boiling rice remove inorganic arsenic? How long to cook the rice to get rid of arsenic? Does rinsing rice remove
Yeah.
arsenic? All of these questions. So if you can touch on this, will be really good.
Yeah, as I mentioned before, you can rinse the rice. It doesn't remove a lot of arsenic. The best is to
When
soak
you
it
boil it,
overnight. You soak it overnight. If you boil, well, there are different traditions of how to boil, how to cook rice. Quite often it is used, so 1 cup of rice, 2 cups of water. So basically, when all the water is evaporated, then the rice is ready. Yeah, ready with this. Then of course, then you don't remove anything. It's not boiling off. But if you cook it in excess water, which costs, however, more energy, you boil more water, and then you have excess water, then you can actually decant the water. And in the water, there's a lot of arsenic there. So you can remove it from this. Not fully, but you can remove a large part. So we investigated it 10 years ago with all different types of water, rice ratios and so on. Yeah.
Yeah. Some of the last question for you, because I know that you have to go to another meeting. Is arsenic in every rice present? You said that it's found in the bran of the rice, but there are different types, like very dark variety, red rice, and you said basmati, jasmine. There's also this wild
rice, yeah.
dark, almost black rice.
It is basically in all rice samples we measured arsenic. However, what is important is if you want really a rice which is very, very low, you go for upland rice. So that means a rice which is grown dry. And all the African rice is usually also drawn, they want to save water, and they have a wetting and drying procedures. And when you do it right, then also the arsenic level is lower. So upland rice, for example, here in Austria, we have now upland rice is one of the lowest we have ever measured. But if you have paddies, so normal paddy fields, then the arsenic is usually much higher. And the reason for this is the arsenic chemistry changes from aerobic to anaerobic, and it has to do with phosphate uptake. So, but how can you distinguish this as a consumer? This is quite difficult, yeah. Quite difficult to do. So in general, long grain rice, American rice is still quite high. But as I said, if you are in a country which have, like here in the EU, when you have a maximum limit, and this is now 5 or 6 years into place, then you can be sure that the levels are lower than the legal limits. And now the EU even lowered it down from 200 ppb to 150. So that's a good sign. So the quality of the rice gets better in terms of the arsenic contamination. But in quintessence, basically every rice contains arsenic, but there are some areas where the rice is lower, and there are different reasons for this, but it's a different talk by itself.
Thank you very much. This was Professor Dr. Jörg Friedman. Thank you for participating in our webinar. And we'll continue now with our second speaker, Dr. Michael Markovic, who is Associate Professor at Graz University of Technology, Austria, co-chair of the GHI Chemical Food Safety Working Group, and a chair of the GHI Supervisory Council. Michael Markovic is a trained biotechnologist who was working for some time in the pharmaceutical industry, developing fermentation processes for antibiotic and other bioactive substances. He is now professor in food chemistry, and you can find more information on the website, GHI website, about Associate Professor Dr. Michael Markovic. Thank you for being with us, and over to you.
Thank you, Diana, for this very kind introduction. So the question is, oil oxidation. Why do we consider oxidized oil as toxic compounds? What was the reason? And especially why are we, or my group, why are we interested in the oxidation of oils? There are some publications you find around the world that there is a certain health risk associated with the uptake of fatty acids, of oxidized fatty acids. And this is, for example, this is mentioned in one of the publications here, and it means that excess uptake of fatty acids can be related to colon cancer by epidemiological means. So increased uptake results in a higher risk of colon cancer, for example. This is one of the problems that we face with oxidized fat. And another publication, which is also published recently, was that it showed this pro-inflammatory effect in the liver. And the idea is that oxidized fatty acids can induce this it's called non-alcohol induced liver inflammation. And we have now these two effects. On one hand, we have this liver inflammation. On the other hand, we have the colon cancer as a result of excess uptake of oxidized fatty acids. So my group is interested in the chemistry of oxidation of fats, and I will talk about these oxidation processes that occur in the fat, in the oils, during storage or during cooking or some kind of heating processes. When you look at the very basic reaction mechanism, you see here, when you look at the fatty acids, we have the double bonds here, and between the double bonds are methylene groups, depending on the amount of double bonds. But these methylene groups have a reduced bond strength between carbon and hydrogen. And with radicals, you can extract this hydrogen, leaving an electron on the fatty acid here, and this electron can move along the double bonds somewhere, and it takes oxygen from the air. Oxygen itself is a radical, a diradical, and one of the electrons, one of the free electrons of the oxygen, can react with this carbon-centered radical from the fatty acid. And you see oxygen is added here, and this leaves another free electron on the peroxide here, and this peroxyl radical, as it is called, can take hydrogen from another fatty acid and forming the hydroperoxide. Either when we talk about linolenic acid, it can mostly react with this or with this carbon in the fatty acid, in the backbone of the fatty acid. The hydroperoxides are more or less stable, and they accumulate in the oil up to quite high concentrations. And then it is possible that they can react further depending on the structure of the fatty acid which is remaining. As you, for example, see here, we have a peroxyl radical here on this carbon here, and this can react with the electron can react with the carbon here, forming a cyclical structure, leaving one free electron on this carbon here, and this reacts with oxygen and hydrogen, forming the hydroperoxide. So we have this cyclic peroxide, it's called epidioxide, and the hydroperoxy is this part here. So, and this you find it also at relevant concentrations in the oil. Another possibility is a similar reaction, but the hydroperoxide is decomposed, losing a hydroxyl radical, and the oxyl radical is remaining on the carbon backbone, and this can form epoxides. And epoxides are also known that these are quite reactive compounds. The problem in this reaction here is an additional problem because the hydroxyl radical that is formed here because of the cleavage of the hydroperoxide, this hydroxyl radical is an extremely strong radical, and it's one of the strongest radicals that we have in our living environment. So you see we have the methylene group between the two double bonds here and related to the number of methylene bonds. So we have the oleic acid, we don't have it. Linoleic acid, we have one methylene group, linolenic acid 2. This is arachidonic acid, we have 3. And this is from fish oil, the docosahexaenic acid. We have 5 of these methylene groups. And when you look at the relative rate of oleic acid and set the relative rate of oleic acid to 1, linoleic acid is acting or is oxidized 40 times faster than oleic acid. And linolenic acid 100 times faster, and arachidonic acid and also the higher unsaturated fatty acids are even faster oxidized. And this is why we have to take especially care of the highly unsaturated fatty acids, what we think are very healthy, because we need it for different metabolic situations in inflammation. We need it for prostaglandins, for leukotrienes, and so on. And further metabolites, so fish oil, which should be extremely healthy, can also be very dangerous because it oxidizes extremely fast. We analyzed the oxidation mainly with liquid chromatography MS because this is a method of choice. We can analyze the triglycerides easily, and with the fragmentation pattern we can identify the position of oxidation. We also can identify which fatty acid is oxidized and on which place within the triglyceride. We could also identify which fatty acid is located in which position of the triglyceride. You see here the masses from palmitic acid to arachidic acid. They are all easily distinguished. You don't need high resolution MS for these experiments. So when thinking in MS dimensions, is a very simple analytical method. And what is also interesting is that you can use a very few, very simple rules to identify the position of the fatty acid. And this is mentioned here. So fatty acids which are occurring at low concentrations in the oils, these fatty acids are normally bound at the position 3. In position 2, you normally don't have, or you don't, in plant oils, you don't have palmitic acid. It's also very helpful. And when you look at the fragmentation pattern, the position 1 and 3 is easily fragmented, while position 2 is more stable. And the fragmentation occurrence is much, much lower, position 2 compared to position 1 and 3. So 1 and 3 are equivalent, and we have the less abundant fatty acids in the position 3. And by this means we have practically all our triglycerides that we identified in the plant oils. We could denominate the position of the fatty acids in the triglyceride. For analytical, when you look at the analytical parameters here, you see we use for the analysis of the triglycerides, we use a C18 reversed phase column. What is extremely helpful for good separation is to add some isopropanol to the methanol. It's very hydrophobic separation, and isopropanol gives some more selectivity to the column. Ammonium acetate is added for ionization in the MS flow rate, and we measure in the positive mode. So here are some results shown, which are calculated and measured. You see, for example, palmitic acid, linoleic acid, palmitic acid. This triglyceride, PLP, has a protonated mass of 831. Then we have the adduct of ammonia or the adduct of sodium. And then you can see the fragments here, meaning that the linoleic acid is fragmented, which gives a product, remaining product of 551. Or if one of the two palmitic acids it fragments, we have a mass of 575. And it's a similar triglyceride with palmitic acid, linoleic acid, and oleic acid. We have a molecular mass of 857, and according other ions, and the linoleic acid, oleic acid diglyceride has a mass of 601. Palmitic linoleic is the same, and the palmitic oleic acid is 577. So this is what you have to, one example, what you have to expect when you do the chromatography and look at the mass spectra of the respective triglycerides. Chromatography looks like this. In the MS, you see the trilinoleic, oleic acid, OLO, the triple O, and the oleic acid, stearic acid. So this is a separation on the C18 column using the MS detector. You can also analyze it with a refractive index detector, which is unselective, and you get practically a signal for every compound that you have. So you dilute the oil in the eluent and analyze it. And what you can see here, for example, in corn oil, we have the region here. The region in front is are the monoglycerides. This region here are the diglycerides, and this is the region of the triglycerides. And you can see that there are distinctive peaks which are separated and which can be analyzed by MS. Of course, corn oil and plant oils are quite simple to analyze. With fish oils, you have a little bit problem with this method because of this huge heterogeneity in the fatty acid composition. But you see, we have all three oils here: sunflower oil, rapeseed oil with the monoglycerides, the diglycerides, and the triglycerides here. Good. Let's have a look at the mass spectra of one of the triglycerides here. We have the trilinoleic, and we see the 879, which is the protonated ion. We have the ammonium adduct with 896, and we have the sodium adduct plus 23, so we have the 901.7. This, in fact, is the molecular ion. And when we do the fragmentation and we lose one linoleic acid, we get one single fragment molecule with 599. Because any loss of any fatty acid always gives the same fragment after the fragmentation. This looks a little different when we look at the mass spectrum of the palmitic oleic triglyceride. You see we have the molecular mass here with the protonated ion. We have the ammonium adduct and sodium adduct, and then we get two fragments. We get one fragment which gives the PO, and we have one fragment which gives the double O diglyceride with 603 and 577. So these are very simple calculations. What is remaining, because we have this table with all possible compositions, and we just identify the mass and see which fragment this is. So the identification of the triglycerides is in fact simple, with very simple mathematics. Similar here with linolenic acid, so oleic acid, linolenic acid, linoleic acid, and we see a fragment or three fragments which are here. We have the linoleic linolenic diglyceride with 597, the oleic linolenic with 599, and the oleic linoleic with 601. And you see, what you do not really see is the distribution, and which shows that you have a higher fragment, a higher concentration of fragment where is more stable, which means we have the linolenic acid here in the center, which is not easily fragmenting. Same here. Again, we see the molecular ion peak plus the fragmentation of the triglycerides. So when we look at the PLO triglyceride, we have the LO, PL, and the PO diglyceride, giving this very typical pattern of the triglyceride fragmentation in the mass spectrum. And in this case, we have the fragmentation in a single quadrupole and no isolation of the ions with later analysis. So this is only in a single quadrupole measurement. Yeah, and the idea was not to analyze the triglycerides or identify the triglycerides in the oil, because this is, of course, quite well known. But when we look at the fragmentation pattern here, anyway, we can see that we get a loss here, which gives a shorter fatty acid, or we lose the fatty acid from the triglyceride here, and then we have a structure like this remaining, which survives DMS and which is measured then. So by this way, we can identify which fatty acid fragments and in which position it fragments. And now this picture shows what's happening in oxidized triglyceride. And I have one example here that you find. I mean, this is from our analysis that we made. You see this endoperoxide here on this position here, and one epoxide on this position here. And then, of course, we have to analyze the fragmentation which is occurring. So we have a loss of oxygen here, Normally, loss of oxygen is some kind of elimination reaction of water, so -18 is lost, or in this case is only one oxygen eliminated, so we have -15 here. And in the next step of fragmentation, we lose the fatty acid here, remaining in a structure which has either one epoxide, or it has the two epoxides, depending on which fatty acid is lost, or we lose the fatty acid with the epoxide. Then we have the diglyceride with the endoperoxide. This can fragment. So here is no epoxide. Here's the epoxide, and the oleic acid is eliminated here, and so on. So, and the further reactions. And by this way, all the peaks that are occurring in the MS that you see here, all the peaks can be denominated and can be attributed to a fragment which explains the original structure of the oxidized triglyceride. Good. So this is a lot of hard work calculating, not difficult mathematics, but a lot of hard work calculating. And I will come to the conclusion here. You see, we have the, I have shown that the chromatographic separation is easy. And it's easy to handle. The eluent is very simple: propanol and mixing of isopropanol and methanol, and this can be analyzed in very high efficiency, especially with the modern columns, core shell materials, and so on. So this is a very good method of separation. We can determine the positional isomers because of the fragmentation pattern. Although it seems that the mass is not selective for the position and isomers, but in this case it is, because we have a different probability of fragmentation at different positions, and we can say which fatty acid is located in which place. And we can also identify the hydroperoxides, we can identify endoperoxides, epoxides, the double bonds, alcohols, whatever compounds are occurring. Aldehydes are analyzed by a different method, or they can be analyzed using gas chromatography, which is very nicely established for the aldehydes. So we can identify all the products that are occurring in the fat. And what you may have seen in the beginning was that the reaction is not very difficult to understand, but the mixture of products is extremely complex, because we have the three fatty acids, we have the different oxidation products, we have the different positional isomers of the hydroperoxides, of the endoperoxides, epoxides, and so on. So we get a huge and very heterogeneous pattern of the fatty acids, of oxidized fatty acids. Yeah, and the last point, which is not mentioned here, is the most difficult part, because we know that we have a health risk associated with the uptake of oxidized fat. Although we have done a lot of work of identifying oxidized fatty acids, oxidized triglycerides, we have no idea which of the compounds are responsible for this health risk. Is it the aldehydes, like for example the hexanal or heptanal, or is it the peroxides, the endoperoxides? So in fact, we have no idea how we can relate our results with the results of the toxicologists. And by this, I want to thank you for your attention, and if you have any questions, you are welcome to ask. Thank you.
Thank you, Michael. This was a really very complex method that you tried to put it into a very kind of mild form so we can all comprehend. So what are the factors affecting oil oxidations?
Yeah, this is very simple. It's in fact this is the presence of oxygen that we have in the oil, and on the other hand, if oil is exposed to the sunlight, then it's the sunlight, of course. So we have to protect oils from the sunlight. We keep it in the refrigerator, and if possible, we keep away the oxygen, but this is not possible. Oxygen has a very good solubility in water. It has very high solubility, much higher than, for example, in water. So we get high solubility, and oxygen is always present. And with this, we cannot really protect it. We could add, for example, antioxidants to protect the oil, and this helps if you keep the oil in the dark, maybe at lower temperatures, so in the fridge, not exposed to the sunlight, in dark bottles, and when you add antioxidants, you can protect it at low temperatures. At high temperatures, so cooking oil, for example, when you cook at 100, 120, 150 degrees or 180, we have always seen in all our experiments, we have seen when you add antioxidants, you get a pro-oxidant effect. So it always is oxidized faster when you have added antioxidants. This is, I don't know if it's problematic, but this is some kind of observation that we have. Yeah. So when you use oil for cooking, it's better to use saturated fat, like coconut oil, for example, which is more stable than oils which are highly unsaturated, of course.
Yeah, and this is related to my next question. How do we keep oil from oxidizing? You said what type of oil to use and to keep it away from light.
Yes, away from light in dark bottles.
Cool. But what else?
You keep it closed.
Are there anything that is helping to the stability?
Yeah, you can add antioxidants. You can add like vitamin E, rosmarinic acid, or ascorbyl palmitate, for example, can be used. So this helps during storage at around room temperature or maybe 40 degrees, but it does not help at temperatures above 70, 80 degrees. So during storage, you can protect the oil. At higher temperatures, it's difficult.
So oxidation could be described as a degradation of good oil to gone bad. So why good oils turns bad? This is another question we have from our audience.
Yeah, the problem is that we have the double bonds in the fatty acids, in the triglycerides. We have the double bonds, and this is a big issue. Yeah, that's the reaction. So we have the oxygen and the double bonds and the methylene groups in between. This is the reaction. It's difficult to answer why oils do that. They're just there with the oxygen, and then it's oxidized. So if we keep the oxygen away and the light, which induces radical formation, if we keep away radicals, I mean, normally it's oxygen, but in presence of copper ions, for example, or iron 2, iron 3, this reaction is also inducing oxidation. So during cooking, maybe stainless steel is more helpful than just cast iron pottery, something like this, or copper is not so much used anymore. But yeah.
Yeah. Are there any specific food that our consumers and in terms of our audience here will have to be careful when they are cooking or using for creating other foods, what they have to be aware of.
Yes, I mean the stable oils or fats are the saturated, okay, or olive oil, for example, which has only more or less only oleic acid. But when you prepare foods with highly unsaturated fatty acids, like linolenic acid, so let's say with omega-3 fatty acids, these are much more, it is much easier to oxidize the omega-3 fatty acids because they have more double bonds. And so with this, you have to be much, much more careful. Otherwise, they start oxidizing. And the other problem is that it is not the health issue only, and they smell very bad when they are oxidized.
So this is how they can recognize that is wrong to consume.
Yeah. And anyway, my suggestion is very simple. Do not use oxidized oils. As soon as you smell the oxidation, this typical rancid aroma, which arises from the aldehydes, it's better not to eat it.
Thank you very much for this participation. Thank you, Professor Dr. Michael Markovic, and thank you, Professor Dr. Jörg Feldman, for your interesting presentation. Both inorganic arsenic and oil oxidation are issues that are really needed special attention as potential toxic compounds in food. Thank you for having the time and joining us. Massive thank also to our audience for joining us today. I hope you found our webinar informative and that it added value to your previous knowledge. I would like also to extend my gratitude to our webinar team. Again, thank you, Dr. Nicola Stanley, our GHI Communication Director, and Dr. Gerhard Schlenning for their invaluable contribution during the GHI webinars. Thank you for your attention, and goodbye from me, Dr. Diana Bogieva. See you at our next webinar, which will be in June, 7th of June and 10th of June. Till then, keep learning, smiling, stay safe, healthy, happy, and curious. Goodbye.